welcome everyone to the first event of 2021 for CSE alumni and friends. I know that today's event will set a great tone for a wonderful year of programming to come. My name is Eric Snewstad. I serve on the engagement committee of the CSE Alumni Board, which helped organize this terrific event and also organizes many other in-person and remote events throughout the year. For me, this is a fun event personally because I work as a patent attorney, and so I have some interest in seeing technologies that come out of the college. And also the firm I work at, Frederickson and Byron, has been a sponsor of the Minnesota Cup's Life Sciences, Health, and IT division, and Blue Cube Bio emerged from that division and won the grand prize in 2020. So this will be very exciting. So without further ado, I'd like to introduce our esteemed speaker for today's presentation. Dr. Allison Hubble is a key opinion leader in preservation with more than 30 years of expertise. She is a professor of mechanical engineering and director of the Biopreservation Core Resource, which is a national resource in biopreservation. Allison is founder and chief science officer of Blue Cube Bio, which stems from 7 years of federal funded research led by Allison. So please join me in welcoming Dr. Allison Hubble.
Thank you very much, Eric. It's been fun to watch people join the conversation, and I see one familiar name in the chat already. A shout out to Joe. It's been a while since we've talked, but it's good to see your name. I'm going to go ahead and share my screen here and put it into presentation mode here. So it's really fun and a great opportunity for me to talk to you about a little bit about our journey. And just so you know, our journey is not complete. We have a long way still to go, but we have learned much and had great adventures along the way. So my technology is relevant to the field of cell therapy. And as the name implies, cell therapy is a form of therapy in which cells are used to treat disease or injury. And in this slide, we see a partial listing of cell types that are used therapeutically. And what you'll note on the right-hand side of the slide is there's a much longer list of diseases. So what happens is cells can be used to treat more than one disease. A given cell type can be used to treat more than one disease, which is exciting because usually one medical device is usually treating one specific medical disorder. It's important to note that part of the reason why I got involved in research in this area is the University of Minnesota is a world leader in the area of cell therapy. We have a GMP cell manufacturing facility on the St. Paul campus. That facility manufactures cells for use therapeutically and sent to patients all over the country. So this is something that University of Minnesota brings and is a world leader on. So what we're looking for, our goal, our long-term goal is to make cell therapy a standard of care, a common method of treating disease or injury. And right now we have pharmaceuticals, most of us know drugs, we have biologics, vaccines are a biologic. Medical devices are another form of therapy. And our hope is that cell therapies becomes that fourth pillar of therapy. Now, in order to achieve that outcome, there has to be the ability to manufacture the therapy. There has to be a regulatory structure because the FDA needs to approve this therapy. There needs to be a way that insurance companies can reimburse producers of cell therapies for the cost of those therapies. And it also has to be adopted by physicians. Physicians have to turn around and prescribe a cell therapy for the treatment of a given disease. So it's important to understand where my work fits into this structure of making cell therapy a standard of care. It's a critical element within the manufacturing infrastructure. And it also enables adoption as a standard of care. If we can transport the therapy to a doctor's office or a battlefield or someplace like that, then it will be adopted as a standard of care. So let's talk about the supply chain for cell therapy. It's unlike any other kind of therapy. It's very different from medical devices or drugs. Typically, cells are collected from a living donor, could be you or me or someone else. It's then transported to a manufacturing facility. You can't just process the cells anywhere. And it goes through a manufacturing process, at which point it's then transported to wherever it's going to be used. And it could be given back to me if they took the cells from me, and that is called an autologous therapy. Or it could be given to someone else. Maybe I'm being generous and I'm giving it to Eric. So that is called an allogeneic therapy. So it has to be sent to the patient. Now, this supply chain is completely different than a medical device. Medical device, you have the raw materials coming into a factory. It may go to a sterilization plant, and then it goes to the hospital where it's implanted. So the supply chain for cell therapies is completely different than other forms of medical therapy. And the other wrinkle to this supply chain is that the cells have to remain viable and functional all along that supply chain. And preservation is the technology we use to stabilize the cells all along the supply chain. So not surprisingly, there have been companies and therapies that have failed because they have not addressed the supply chain issues. And I'm going to talk about two examples. One is Provenge, which is a dendritic cell therapy. They had a very short shelf life. It has about an 18-hour shelf life. In fact, they built a factory next to Atlanta Hartsfield Airport so they could minimize the amount of time between completion of manufacturing and actual shipment of the therapy in an airplane. It's important to know that in more than one occasion, the patient didn't show up for their therapy and the therapy had to be thrown away, and the company eats that cost. And as a result, this therapy and the company failed. Example number two is mesenchymal stromal cells to treat graft versus host disease, and that product is called Prochymal. And it had made through phase 1 and phase 2 clinical trials. They got to the phase 3 clinical trials, and they could show no therapeutic benefit to their cells. In other words, the outcome of patients with and without cell therapy was no different. So the phase 3 clinical trial failed, and usually that doesn't happen. And the company didn't exactly give a reason why, but other individuals who've done research in the area hypothesized that poor methods of preservation meant that the cells were not clinically effective, and as a result, the clinical trial failed. So it's important to understand that we've already had poster children, in a matter of speaking, as to why supply chain issues are critical and can result in failure of a very important, potentially transformational medical therapy. So about 7 years ago, I was sitting in my office. It was another lovely winter day like we're having today. And I said, how do trees survive Minnesota winter? Because I can see it. I'm in my office right now. I can see a tree outside my window. And the answer to that question is they express molecules that protect the cells against the cold. And those molecules are known as a category as osmolytes. So it's important to understand that strategies that trees use are also used by a variety of other biological systems. There are small animals like the tardigrade or water bear, the resurrection plant, and brine shrimp cysts, each of which use combinations of osmolytes in order to stabilize themselves against environmental extremes. A tardigrade lives in the soil and can actually be dehydrated for up to 10 years and come back rehydrated. I know the resurrection plant can do something similar. Brine shrimp cysts use osmolytes to stabilize themselves against high salt environments. Now, it's not just nature, it's us. Our kidneys collect and secrete urine, and that is a very adverse environment for cells in the kidney. And our kidney uses a combination of 5 osmolytes to stabilize itself against the adverse environment that results from urine collecting in the kidney. So it's important to understand that nature has been using these molecules to stabilize cells in all kinds of different organisms, and that it's never just a single molecule, it's combinations of molecules. So we put forth a hypothesis and we said, hey, combinations of osmolytes can stabilize cells without the use of dimethyl sulfoxide, which is a chemical commonly used to preserve cells. And they said, okay, this is intriguing. We'll give you a little bit of money. And with that little bit of money, we proved kind of some of the little bits of the hypothesis that we had. And they said, okay, you did a good job. We'll give you a bigger bucket of money. And with that bigger bucket of money, we were able to really get traction in this area. And we found that literally hundreds of combinations of osmolytes provided a protective benefit, that in fact, the synergistic or combinational effects of the molecules were strong and ubiquitous. Once again, that's exactly what we would expect from nature. Exactly. Nature would never come up with a single combination of molecules that helps to stabilize cells. They would always have families and numbers of molecules because not every organism is going to be able to express the same molecules. So once again, we are seeing a story that is consistent with that seen in nature. It's important to understand that these osmolytes are naturally occurring, non-toxic, common, and in fact, most of us eat the molecules that we use to stabilize cells every day. So these are naturally occurring and safe molecules. So we had to unpack this, however. We have to understand on a molecular basis why these molecules work. And in the left-hand side of the slide, we have the capability at the University of Minnesota. We pioneered this measurement technique. We can take cells down and freeze them and then image them in the frozen state using Raman spectroscopy, which is a laser that shines on the sample, and the way that it reflects from the sample reflects the chemical bonds present. So we can actually identify different molecules in the sample. And in the image I'm showing you here, we identify ice. We identify amide 1, which we are saying is a marker for the cell itself because it's a common chemical found in proteins, and cells are a lot of protein. And then we also looked at the presence of sucrose, which is one of those osmolytes that we're using to stabilize the cells. And what we found here is the sucrose wasn't going inside the cell. We could show it in unfrozen solution surrounding the cell. But what it did do was it actually integrated itself into the cell membrane. So one of the ways that these molecules stabilize the cell is by interacting with the cell membrane. Another thing we looked at is there's clearly interactions between molecules. And we use some statistical modeling to understand that, and that really didn't make a very good figure, so I didn't include that. But basically, we use statistical models to prove that there were interactions between molecules. But in this right-hand side figure, we're looking at differential scanning calorimetry, a very, very common experimental technique in which we're looking at the amount of energy released when we freeze the solution. And the area under the curve tells you how much ice is present in the solution for a given composition. And what we found is that sugars and sugar alcohols interact with each other, and they interact with water to prevent the formation of ice. So here we are starting to really unpack and understand the interactions between these molecules and other critical structures, one of which is the cell membrane and the other is water. Okay, now we went forth and we established our ability to use these combinations of osmolytes on two different cell types. One is mesenchymal stromal cells, and those are cells found in our bodies right now. They help to improve healing and moderate the immune system. And then we have induced pluripotent stem cells, which are a genetically modified cell that are being used as a basis for creating other cell types that can be used to form tissues and treat disease. So both of these cell types, we have proven that we can preserve them with combinations of osmolytes. And then we've done other work with T cells, which are immune cells in your body, and we're working on smooth muscle cells and a whole variety of other cell types. So once again, this platform is applicable not to just one cell type, but multiple cell types. Okay, so there's another kind of, so this is for the nerds in the audience. It's important to understand that when we talk about trying to optimize a mixture of multiple components, the normal way of doing it would have been you put in combination A, you freeze the cells, you thaw them, and you determine post-thaw viability. And we call that the matrix of death because that's the method that a lot of cell types were, preservation protocols were developed for a lot of different cell types. And in fact, some of the earliest work on preserving red blood cells using freezing, it took people in the Naval Research Lab a decade to come up with an optimum protocol. But as engineers, we know we can't do that. We have to be able to come up with optimization that is quick, efficient, and doesn't require a lot of resources. So we chose to use differential evolution to optimize. We didn't know how the variation and viability would be with composition. But in the right-hand part of this slide, this is some arbitrary population. So what we do is we pick a random distribution of conditions that span that population, and we do a test and we determine what the post-thaw recovery is. We then insert that outcome into the algorithm, and the algorithm says, okay, you did that, now try this. So we tried the next generation. And the next generation after that, we put in the post-thaw recovery, and then we get the next guess. And what you see with time is you start in a relatively small number of experiments to define that parameter space and come up with an optimization strategy. So we said we're going to use differential evolution algorithms, and we're going to use that to optimize our freezing protocol. And in this next slide, what you see is the outcome of these different experiments. So basically in 8 experiments, instead of thousands of experiments in a decade of time, in 8 experiments, we optimize the post-thaw, the composition of a cryopreservation solution containing 3 osmolytes. And we were able to get better post-thaw recovery than the gold standard. And we had a functional assessment of viability. So here's our technology platform. Our technology platform means that we can take molecules from nature, we can combine them in a way that results in high levels of post-thaw recovery and function, and we can do it in a really efficient manner. We can figure out this in a relatively short amount of experiments. So that allows us to develop protocols for different cell types and then optimize that rapidly. So we understood how these molecules work. We understand why they work. And this allowed us to get very generous claims from the patent office. So we have an issued patent and additional IP that is in prosecution. So good science can lead to good patenting. But what happens now? We know we can do this efficiently. We know we can preserve several different cell types without DMSO. We have patent protection, but really, is this a product? Is this a business? And one of the first questions you have to ask is there a product market fit? So now we're moving out of the realm of research and into the commercial realm. So first top was the MNCOR program. That is a local I-Corps program funded by the National Science Foundation. And there's a series of workshops that we go through, product market fit, customer discovery. Yes, we have to go out and talk to people. What a concept. We have to talk about the pathway to commercialization and market assessment. Now, the program also provides just a few $1,000s that allow, let's say, someone to travel to a conference and do some of the interviews. The three figures at the bottom of this slide, the first is Carla Pavone. She is the director of this program and has provided tremendous mentorship and feedback and all kinds of wonderful things to us on their voyage. And the other two are the PhD students who participated in the Minnesota, in the MNCOR program, Ray Lee and Katie Hornberger. So we went through and interviewed 30 people, which you think, wow, that's a lot of people, but you've only just started. And that initial feedback was positive. It said, yes, there's a tremendous need to improve preservation and to get rid of dimethyl sulfoxide, but we needed to probe further to understand the specific pain points and the specific value proposition that this particular technology would afford. And as a result, we went on to the National I-Corps program. In order to do that, we were given a grant by the National Science Foundation for $50,000, and that $50,000 could be used for things related to commercialization of the technology. And we used part of it to go to the National I-Corps program in lovely Newark, New Jersey. Yes, we were within shouting distance of the airport. And for those of you who have traveled in that area, it's really lovely. But we spent most of our time in the hotel. We had exercises to do, we had coaching and one-on-one coaching and instruction. And we had also arranged for interviews in the greater New York City area. When the course finished for the day, we would spend the afternoon and early evening taking Ubers into Manhattan, and we interviewed both companies and academic cell therapy labs all over Manhattan. And within the week that we were at Newark, we completed about 20 interviews, and it was quite the adventure, because we would spend an hour with someone and then madly jump in an Uber and drive to the next interview. And once again, the point here is to talk to as many people as possible, understand the obstacles that they're facing and the obstacles potentially to the adoption of your product. So after our 1-week course in Newark was over, we then took the $50,000, what was left of the $50,000, and expanded outside of that. And the little stars will indicate all the different locations where we either visited in person or with whom we talked on the phone, because we did both phone interviews and in-person interviews. The stakeholders included cell therapy companies. It included hospitals or cell therapy labs. It included federal agencies. We actually went to the FDA, not for anything related, but just to kind of get the perspective from a federal agency. And we went to other federal agencies as well. We completed over 126 interviews. This is in addition to the 30 we did with the local MNCOR program. And all of these experiences help you develop, help you understand the major pain points. So what we were able to do with this large number of interviews was understand the role that adverse reactions to patients, because DMSO causes adverse effects when infused into people. It also causes complexities in the actual manufacturing process. So once you add the DMSO, which is the conventional method of preserving the cells, you have a really short period of time before your cells have to get inside the freezer. And all of this adds complexity and difficulty all along the manufacturing process. And the end result is large percentage of cell losses. Roughly 50% of the cells or greater are lost. And since these cells are very expensive and precious and in general can't be replaced, that is something that threatens the viability and the clinical potential of the product. So what we are seeing is our product addresses these pain points, and there is a need to improve the field. So the other thing we know is that this technology is disruptive, that there have been little changes in the field of preservation since the 70s, and that we are causing a transformation in the field because we are using common, naturally occurring, and safe molecules to preserve cells. And we can rapidly optimize for a given cell type. This is somewhat staggering to the community because what they have been looking for is one solution that can preserve every cell type. And for those of you with a little bit of a biology background, you know that different cells have distinctly different biologies, and it's extremely unlikely to have a single solution that could preserve every cell type. So we know that, we know we have need for our technology. We also know that it's a disruptive technology, which has implications for moving to market. Well, pandemic time. So we were flying all over the country, interviewing all these people, and then the country went into shutdown. And so we were literally grounded. And we ended up finishing our program over the internet. And what's interesting is NSF is now holding the programs virtually. They're not going to hold any more in-person programs. We thought, hey, why not start a company in the midst of a pandemic? What's the worst thing that could happen? And part of that motivation is it became very clear that there was strong market pull for what we were doing. And also some of our products were directly related to the treatment of COVID. Specifically, our method of preserving mesenchymal stromal cells is relevant to the use of those cells to treat COVID-related lung injury. So we took a leap of faith and incorporated in the midst of a pandemic. And it's kind of funny because we spent all this time together through the National I-Corps program, and since incorporating, we can't be in the same room together. Or actually in the backyard. We were at one point in the backyard outside together, but it's been kind of an adventure. So one little slight pivot here in that there is a funny story about the origin of the name. It's Blue Cube Bio. And when my son, who's pictured on the right, as you can tell, he's a grown-up now. When he was in preschool, I had to go to a parent-teacher conference. And they said, you know, Alex just looks at things and he sees things in such a different light. And she picked up a blue wooden cube that was sitting in the room. And she said, if I hold this up to the class, everybody else in the class says it's square, whereas Alex will say it's blue. So the name Blue Cube Bio is meant to say and meant to encapsulate the concept of taking and looking at something, in this case, preservation, and seeing it through a different lens. So innovation is built into our name, and it's part of what we are as a company. So it came from a preschool parent-teacher conference. Okay, so off to MinCup. Why not go through a business plan competition in the midst of a pandemic? Full disclosure, I didn't want to enter Minnesota Cup. I know, everybody else on the team wanted to. Our list of mentors grew, and on the right-hand side, once again, oh, and I missed this before, Karen Dodson was our mentor for the National I-Corps program, and now we've added more mentors. Martha Sewell, Lee Jones, Pam York. People were tremendously generous and wonderful with their mentorship and their advice to us. And yes, I will admit, in spite of my misgivings, the prize money, it comes in handy, especially when you're a startup. And we did get some additional support from Discovery Launchpad, which is another University of Minnesota. They helped us with some of our financials. So it was quite the adventure. It took a lot of time to go through all the different steps of the competition. And it was also very strange to do it all virtually. But it was a worthwhile adventure. We got a lot of wonderful advice and mentorship and connections with different resources that can be helpful. So I'm going to talk just a little bit, because you've seen a lot of women's faces here, and that's atypical. Most of innovation is brought forth by male innovators. Only 10 to 15% of women are innovators. And this gender discrepancy begins all the way at the invention disclosure stage. And the University of Minnesota has done some important benchmarking to understand where the pipeline gets leaky. We as a team, Blue Cube Bio has faced a lot of challenges in terms of our male colleagues were offered opportunities and deals on rental space or other things that we were not offered. What this has forced us to do is really mindfully and consciously expand our networks, because we're going to have to be, if we're looking for lab space, we're going to have to look at more spaces than other people would have to look at. And we also have been tremendously benefited by men who are invested in helping women move forward in innovation as mentors, funders, etc. So it's been quite the adventure. It's important to understand the University of Minnesota has a commitment to diversity, equity, and inclusion in innovation. And we have programs to actually enhance participation by women and people of color. And so this program is supported by NSF, and it allows us to address gaps in skills, networking, and also to bring together investors so that innovators, women innovators and innovators of color can understand the thought processes and the decision-making that goes on with an investor. And that is extremely important in helping them move forward. This program, we're hoping, will be a model for other programs. In particular, NIH is now interested in enhancing diversity within innovation. And it's consistent with the university-wide commitment to enhancing diversity, equity, and inclusion. So all of these efforts are consistent and reinforcing themselves. And my hope would be that you will see more women and more women teams and also communities of color involved more and more in innovation. And that's important not only for our society, but also economic growth, because there's a very clear, strong business case for diversity. Okay, so back to all these diversions about where the name came from and women innovators. Let's get back to Blue Cube Bio. We're working on brand positioning and our go-to-market strategy, quality systems, our product pipeline, and putting together the team that will help us get to the next phase. It's important to understand that we've had tremendous market pull. In fact, it's almost been like market tsunami. It is actually something that was in some ways detrimental because we've had almost 30 companies contacting us. And when everything becomes a priority, nothing becomes a priority. So part of what we have been doing is trying to develop a method by which we can manage these relationships and make priorities out of certain relationships and put other ones on the back burner. So our market pull is strong, which is good. So the good news is our market pull is strong. The bad news is our market pull is strong. So we have to be able to learn how to manage that market pull. So I've had to, I'm an engineer, and of all things, a mechanical engineer. And one of the things that I've had to do is learn how to network. And when I networked, then I can network again. And when I am done with that networking, I can network some more. So that element has been very different than your typical engineer will have to deal with. I've had to also grow my management and organizational skills because growing a company is a lot different than managing a lab at the University of Minnesota. And I'm still learning the language of business, even though it is ostensibly English. It is a language that I am still learning on, and all the terms and what they mean and why they're important. So I've had to grow a lot along this journey. And one of the things I do is I have a small number of startup CEOs that I call, and I cry and scream or whatever. And one of whom said, look, I'm a rock climber. And you know when you climb rocks that the one thing you cannot do is stop, because if you stop, you're going to fall. And he said a startup is the same way. You have to keep moving or you're going to fall. And so that is what I'm going to leave you with, is that lesson of keeping moving and keeping progressing down your path to commercialization. And I'd like to thank you for your attention, and I'm happy to take whatever questions you have at this time.
Okay. Thank you, Dr. Hubbell, for sharing your work and voyage so far. That was terrific. We now have 10 minutes for questions. And so I'll get us started with a two-part question. First, how have you and Blue Cube Bio leveraged the university's technology commercialization office and programs like the Venture Center and Discovery Launchpad, which you mentioned in your talk? And second, setting aside your personal voyage, how is the university as a whole doing things to help faculty and student researchers bring technologies from the lab to market?
So, it's always interesting because you're right. There's the Discovery Launchpad, there is the Business Advisory Group and the Venture Center and everything. And, I pitched in the Business Advisory Group and interviewed several CEOs, but I didn't pick a CEO from that group. But those experiences helped me understand how to look at someone's credentials, how to understand their experiences. Discovery Launchpad was helpful to us, but we were going through the Minnesota Cup and not really getting ready to pitch to investors. So the path that every startup goes through is different. And so sometimes you get the timing where it works, that you fit perfectly within the university's resources and you can go with it. I will say that the university's investment in the MNCOR and other things like that, wonderfully helpful, tremendously helpful. And that aligned really well with where we were going and when we were going, where we were going. So I have been involved in commercialization of faculty research for over 5 years. And I can tell you, because I was part of a large grant from NIH called MinReach, and it was a $6 million grant that helped faculty in the medical area commercialize their innovations. And I can tell you the university has, over that period of time, really transformed the resources, the mindset, everything about commercialization. And I only see that continuing. There is tremendous traction in terms of both grad students, postdocs, and faculty who are interested in commercialization. There is increasing diversity of resources that can be brought to bear to help someone move along. That being said, there can always be more. I don't think we've even come close to reaching saturation. We still need investors. We still need startup CEOs. We need other aspects of infrastructure that will grease the wheels for these the faculty innovation to move forward. Keep in mind the research enterprise at the University of Minnesota is a $1 billion enterprise. And so there is tremendous potential within that $1 billion of research to translate the research knowledge into products and services, whatever, to advance human good and improve the economy of the state of Minnesota.
That's great. Let me go to questions from our audience, and I'll start with one that is kind of a few different questions, and you can object on this being a compound question, but since I already did that, I guess I can't. All right, so how did you identify your market segment? Was it driven by interviews? What kind of questions did you ask in these interviews?
Good question. Okay, so part of my agenda in focusing on cell therapy is I have 30 years of experience, so I understand the market and many of the players. Now, that being said, the cell preservation total market is a roughly $2 billion market, and I'll talk about some other segments. So one segment could be classic biotechnology like CHO cells. That's clearly an HEC-293 or whatever the number is cells that are used for expressing recombinant proteins. Another potential market is biobanking. A third market is, and there's a lot of growth and interest in this, clean meat, where instead of raising an entire animal, we actually culture meat in a petri dish. So there's lots of different market segments. I will tell you that the interviews helped us understand how much the preservation issue was limiting growth in cell therapy. So we knew once again that was a very strong market pull. In other words, our ability to overcome this preservation issue will allow growth in cell therapy and therefore drive our value even more rapidly. And what questions did we ask? That was a voyage of discovery because, you know, some people were really forthcoming when they had problems, and other people weren't going to tell you that everything's okay, nothing's a problem. And you would have to come about things in an indirect way, right? You'd have to say, well, you know, what kind of timeframes are you looking at for, you know, completing the manufacturing process and getting the sample into a liquid nitrogen storage? Or, you know, what kind of cell losses are you experiencing along your manufacturing, and where are those cell losses? And so with some people, they were like, this is a real serious problem. This is what we see. We see this loss in viability. We can't get the product out of the GMP suite and into the liquid nitrogen dewar in time. Other people, it was just extremely indirect. So you had to adapt. You couldn't have a fixed script. And the more interviews you did, the better you got with kind of navigating people's process for actually being forthcoming about what their problems were.
Great. Great. We have a lot of good questions. How about this next one from our audience? You mentioned that your product is disruptive to the market. Although you've had a lot of inquiries, are you seeing resistance or reluctance by some potential customers to try your product because it's so different?
So yes and no. The yes is because in cell therapy, what they want is they want you to have regulatory approval or the proper regulatory filings. And we're actually in the process of addressing those issues. So I think they're looking for any solution, and it's not the molecules and that they're naturally occurring. They want us to have created that regulatory structure so it's plug and play. So they can just order the sample, determine if it's going to work for them, and then start putting it into clinical trials. So they want us to grease that runway completely, and we're in the process of doing that. So that was an obstacle that we hadn't anticipated completely, but we have a clear path. We have a subject matter expert who is helping us develop an approach. He used to be at the FDA. He's aware of my work, aware of the product, and very, very helpful.
Oh, great. Good question, great answer. Here's another one. What is the product goal for Blue Cube Bio? Is the plan that the end product would be the therapy, the cells and the solution to preserve them, or that the end product would be the preservation solution that would then be sent to cell manufacturing companies to integrate in their products?
The second. So in FDA speak, we are an ancillary material that is used in the production of a cell therapy. So our product does not have a therapeutic effect. So yes, the end result would be little bottles of solution and a method of use. The other thing we found out is you can't just give people a bottle of solution. You have to give them a very detailed protocol for proper use in order to get the desired post-thaw recovery that we receive. So on some level, it's a little more high touch than most cell therapy or most ancillary material companies, because we have that method of use.
There are so many interesting things in your talk, and the questions reflect this. Here's one that struck me too. The differential evolution experiment design model seems very interesting. Can you recommend any how-to resources on this subject?
Storn and Price have a website, and they have the algorithm in like 17 different languages, every computer language you could possibly imagine. So it's open source shareware. It's publicly available. It's been applied in all kinds of different applications, everything from planting of crops to, optimizing cryopreservation solutions. So the good thing about the differential evolution algorithm is you don't have to have a continuous function along the parameter space that you're optimizing. And because we didn't know, we were just like, we have no idea if this is going to be a continuous function or not. So let's just try this and see if it worked. And it worked gloriously. And yes, it can be used for all kinds of different applications.
Great. I wanted to ask a question on the technical side too. One of the many interesting things you talked about is your team's work imaging intracellular ice formation. And I imagine there are some challenges when you're imaging freezing cells like that, but I also imagine this is a pretty powerful tool in your work. And as kind of an open-ended question,
oh, Eric, you put yourself on mute accidentally.
Well, that was just to check your lip reading expertise. Sorry. Let's try it again. That gave me a good dry run. So on the technical side, one of the really interesting topics that I heard you talk about is your team's work on imaging intracellular ice formation. And I imagine there are some challenges when you're imaging freezing cells, but I also imagine this is a powerful tool in your work. And I thought it might be fun to hear you speak a little bit more on that topic, if you could.
So the first images of cells being frozen that were examined microscopically was in the late 70s. And that was just regular light microscopy combined with a temperature controlled stage. And they're really, until we started doing Raman imaging of the cells, that was the principal modality used. And we wanted to do Raman imaging because of the chemical specificity. So for a given image, we can look at all kinds of different signals. So what have we looked at? We've looked at ice, whether it's ice or water. We've looked at where the sugar or osmolytes are and where they're not. We can look and see, like for an immune cell, for natural killer cells, we can look and see where the granules are, and that has the killing activity. So we can look at different biological entities. The big limitation, there's several limitations, but one of the big limitations is signal to noise, because some biological signals, especially at low temperature, are very low. So can we detect it, and can we differentiate it from noise? But the simple reality is there is so much information in every image we obtain that we're not even coming close to mining all the data that we have. And in fact, on Monday or Tuesday, I'm meeting with a student who's going to help us open the next layer of information in those images. Because we've been analyzing maybe a half a dozen different signals, and now we're going to expand beyond that.
Great. I can't help but ask this question. So it's exciting to think about where this technology may end up in kind of 1 to 2 years, 5 years, 10 years, and beyond. Could you speak a bit on the following two things? First, right now and in the more immediate time frame, what are the main or kind of top applications for this technology? And second, as you think further out into the future, what are some other potential applications, and where do you hope things will go with this technology in kind of the longer term future?
So in the short term, we want to transform the preservation of cells used therapeutically, which will enable more people to get those therapies. One of the products that we're working on right now in the lab is preservation of natural killer cells, and it's limiting their use for the treatment of cancer. That's it. This is the ability to preserve the cells is limiting the number of patients who can get this therapy. And that's unacceptable. We need to solve that problem so people get the therapies they need. Beyond that, I'm sorry, at some point I want to at least dip my toe in clean meat. It's so freaking intriguing. And I want to see if we can get traction and adoption in that area as well. And then the third market is biotechnology. I think we could really have a very important effect on the preservation of cells that are used to express therapeutic proteins. Once again, it's addressing these workflow issues. And I think that we could have a very positive effect on improving efficiency overall. Those would be the three markets. Ultimately, world domination is what we're shooting for. So.
That's great. Well, thanks, Dr. Hubbell, and to our audience. Thanks for submitting so many terrific questions. We're running out of time, so I'm going to turn things back over to Joelle for some closing announcements.
Thanks so much, Eric and Allison. That was a really interesting presentation and a great Q&A. Wow, we got so many great questions. Thank you all for submitting them. If we weren't able to get to your question or you want to learn more about BlueQ Bio, I have their web address on your screen, and this will also be included in the follow-up email that I send around with the link to view this recording that I'll hope to send next week. That's bluecubebio.com for more information about the company and Allison's work. If you're interested in more ways to engage with the college, including upcoming events and volunteer opportunities and those types of things, you can always go to csc.umn.edu/alumni to learn more about what we have coming up in the spring and beyond and how you can be involved. I want to close things out by telling you about a couple of additional events we have coming up in February. These are open to everyone, so please feel free to share if you know someone you think would be interested. The first one is coming up on Wednesday, February 10th at 12:30, and it will feature a group of CSE students who are part of the CSE chapter of Engineers Without Borders. And they'll be presenting on their work both locally and abroad to help communities in all kinds of different ways. They're amazing students who do a lot of marvelous work, and we're excited to share that with you. And then this is another program that the CSE Alumni Board is helping us to organize. And then later in the month of February, we have the next installment of our Dean's Lunch and Learn series, this time featuring the School of Physics and Astronomy on February 25th, beginning at noon. I have links on the screen, but again, these will be included in the follow-up email I'll send with the link to this webinar, so no need to rush to write them down now. We'll send you an easy to follow link next week. And with that, I want to let you know you can always contact me with any questions you have at cscealumni.umn.edu. And I want to thank you all so much for making time to join us today and to hear about Allison's amazing work. A big thank you to her. This is a busy time of year getting ready for spring semester to start next week, in addition to all the work that's going on with Blue Cube Bio. So we want to thank her so much for giving her time and sharing all this wonderful information about her research and her startup and where it's going to go next. And a big thank you also to CSE Alumni Board member Eric Snewstad for doing a great job moderating the many questions we received today. So with that, I'd like to thank you all again for being part of our program today. Have a wonderful afternoon.